Hello everyone. Welcome to the new International Mycorrhiza Society IMS virtual seminar series. My name is Patricia Silva Flores. I'm an assistant professor at the UCM in Talca, Chile, and also communications director of the International Mycorrhiza Society. Today, together with Ana Bermudez, a PhD student at the CARS lab of the University of Alberta in Canada, we'll be moderating the seminar. We will be moderating the seminar. But first, we will start the introduction of our dear guest speaker by the IMS president, Dr. Marcel van der Heijden. Once today's speaker starts, Anna and I will be collecting your questions that you might have through the chat, okay? So we welcome once again to all of you. We are very happy for this seminar. And I leave it with you to Marcel van der Heijden for the introduction of our dear guest speaker. Marcel, please.
Yes, thank you very much, Patricia. Also from my side, good morning and good afternoon and maybe good evening. It's really a great pleasure that Francis Martin is today speaking as the first talk for the IMS. So I know Francis already a long time, and I think many of you know him for a very long time. I found out actually that he's 43 years at the INRAE in France, 35 years director, so has a very long history in mycorrhizal research especially. He has been editor for the New Fight since 1991. That's before I started my PhD, so also a very long time. Francis is member of the French Academy of Agriculture. He has also been past president of the International Mycorrhiza Society. At the moment, he is a foreign expert at the Huazhong Agricultural University in China and also visiting professor, Chinese Academy of Forestry, so very active there. And I think everyone here attending, you know his work very well, many very famous papers. For me, probably the most important one, but I'm not sure Francis will agree, but it's the one on 2008 in Nature with the genome of Lacaria, which I think was a breakthrough, and then many more papers, and also already before, but many more papers followed. So really very happy that you have the time to present this work, and we all look very much forward. Thank you very much, Francis.
Thank you very much, Marcel. Listen to you, I'm realizing I'm getting old. Huh? So many years working on mycorrhizae. So, of course, I'm very proud to be the first speaker of this new IMS series. I hope many people will be invited to give a talk to this new series. And as you said, I've been working on mycorrhizae for many years. And in fact, it's almost 20 years now that we have started to work on using genomics for getting a better understanding of the mycorrhizal associations. So I'm going to take the opportunity of that seminar to first invite you all in my office to have a very informal discussion about how we can use genomics and molecular tools for a better understanding of the biology of the mycorrhizal association, but also, I think most importantly today, how we can harness these tools to get a better view of the ecology of these associations. So I'm going to summarize what we've done over the last 20 years, trying to pinpoint a few major breakthroughs. And I will open the discussion and we'll try to provide some highlights or insights on what I see as the future of mycorrhizal research. Again, I think it will be very, very cool and very informal, because it's not giving a talk in the front of my computer is not very like giving a talk in a big theater. I like to move, I like to interact with the audience, and there it's probably very, very difficult. But let's start. And sharing my screen. Okay, do you see
Yeah, everything perfect, Francis.
Oh, okay, good. So let's move from my office to the forest ecosystems. Last week I was lucky enough, I was privileged to sample soil and soil cores and mycorrhizae in this beautiful forest, which is located in the Chilean mountains in the Gansu in China. This is an old growth primary forest, and we are aiming to get a better understanding of what's going on in the microbiome, in the fungal communities in this forest. So I think it's very critical today to get a better view, a better understanding on how the climate change will impact these forest ecosystems. And most importantly, how the climate change will affect the soil microbial communities. So all the work we are currently developing in my team is aimed to that goal, how we can use genomic tools to get to know better how these fungal communities are interacting with the trees and interacting between the different fungal communities, the saprotrophic as well as the symbiotic communities. So our aim here at INRAE over the last almost 15 years now has been to focus on the plant microbe interfaces. So let me see if I can get the pointer. Yes, so we are, the whole team here is working on that plant microbe interfaces. And our overarching goal is to characterize, to better characterize the microbial interfaces, which are allowing the transfer of nutrients, so nitrogen, phosphates, potassium, microelements, but also what kind of information signals are moving through that microbial interfaces between plants, trees, because we are mainly working on forest ecosystems, but also how this association between microbes, mycorrhizal fungi, and trees are interacting, are impacted by their environment. And we have been focusing for many times on the carbon cycling, not only carbon sequestration, but also carbon decomposition and how carbon is moving through the trees, moving through the fungal partners, and moving throughout cycling through the ecosystems. And as many of us working on mycorrhizae, we have tried to define the different fungal communities taking place, living in these forest soils by using metabarcoding. Now metabarcoding has been introduced in mycorrhizal research more than 20 years, so it's really a routine tool to characterize, to survey the fungal communities. Then on a few systems, we have been able to characterize the interaction at the molecular level, at the biochemical level, the mechanisms taking place between these different partners. And for quite a few of them, we are now able to manipulate these systems. We are able to manipulate the gene expression in the plants, in the host plants, and we are able to manipulate the gene expression in the fungal partners. So I think now we have, time is ripe to collect all this information to feed the forecasting models, these models which are currently designed to forecast the forest of the forest, the future of the forest ecosystems. We have enough data now to get insights on what's going on in this so-called black box of the microbial world. So I'm going to focus today on mycorrhizal associations. Of course, you know, most of the people today are experts in mycorrhizal research. And you know that in forest ecosystems, we may encounter ectomycorrhizal association. Amanitas, Russula, Lactarius species interacting with big trees. But we also have arbuscular mycorrhizal association in forest ecosystems. We have ericoid mycorrhizae and orchid mycorrhizae. And in fact, today we know very little about the interactions, the interrelationships between, on one side, ectomycorrhizal associations and the other types of mycorrhizae living in forest ecosystems, ericoid, orchid, or arbuscular mycorrhizae. And I think we can use large-scale genomics, sequencing of hundreds of genomes from these fungi to try to predict their ecological traits. And more specifically, we can use these genomes, genome annotations, genome repertoires, to understand their nutrition modes of these forest fungi. And so everything is here in this short video. We are in an understory in a forest, hopefully an old growth forest with old oaks, old spruces, and the root systems are interacting with extensive web of hyphae. You see there in the soil this web of hyphae, hyphal networks connecting the different plants, connecting mushrooms to roots. And we are many here willing to understand better what's going on. What are these fluxes of nutrients, the fluxes of signals, the fluxes of information which are taking place there between the fungal mycelial networks and the roots. And when the mushrooms are formed, how these networks can feed the mushrooms and then help the fungal mushrooms to disseminate spores and their genetic material. So genomics can be used to understand how the hyphal networks are working. Genomics can be used to understand how these hyphal networks are interacting with the root systems, but we can also use these genomic tools to understand the development of the fungal organisms. And so, together with Marcel, recently we published a 10th review in the Mycorrhizal special issue, and we have tried to summarize our thinking about how mycorrhizal networks, including the common mycorrhizal networks, may act in the forest ecosystems. So on this illustration, we summarize what we expect to take place in the forest ecosystem, in the soils, in the forest ecosystems. We have many networks. We have the roots, we have the root systems, we have the mycorrhizal networks interacting with the roots, we have the networks of the mushrooms. And what we do not know today is how these different networks are connected. What are the amino acids, carbohydrates, fatty acids, hormones, maybe microRNA, maybe signaling peptides, which are shuttling, moving, translocating throughout that networks. So this is really a cutting-edge science, and I wish that the young scientists listen to that talk today are willing to focus on these networks, trying to better understanding how they connect the different components of the forest systems. So I'm going to provide you with three main highlights this afternoon on the evolution of the mode of nutrition in these symbiotic systems, and compare these mechanisms with those taking place in saprotrophic fungi. We had three main aims when we initiated that work about 10, 12 years ago. Aim one was to track the multiple independent transitions between saprotrophisms and mutualisms in fungal lineages. Some teams used phylogenetics for quite a few years to demonstrate that most of the symbiotic mycorrhizal species were deriving from saprotrophic species. So we used large-scale genomics to confirm that multiple independent lifestyle transitions. Then we have used these genomes on a large scale to relate genomic features, genomic traits, to key ecological traits like soil organic matter decompositions. And then, of course, we have used genomes, gene repertoires, to aiming to identify the gene networks which are involved in the symbiosis development and functioning. So a single genome can be used for understanding the biology of these fungi, but also their ecology. And if you want to get more information, you can read that 2 review papers that we have published in INRA Review in 2021, and more recently with Marcel in this Tensor Review published a few weeks ago. Today, I'm just providing a few highlights, a summary of this work. So the cornerstone of that program was to link genes to ecological traits. This was really a proof of concept. Can we sequence genomes throughout the tree of life of fungi that you have there on the right, picking fungal species, saprotrophic, endophytic, symbiotic species in most families of fungi? Can we sequence these genomes, annotate them, compare the gene repertoires, looking for gene acquisition, gene decay, gene duplication, expansion or contraction of gene families, looking at the functional divergence between these fungal species? Can we relate that to their ecology, their lifestyle, their modes of nutrition? And then we selected within that fungal tree of life, we selected species which were ecologically relevant, taxonomically relevant, and belonging to different lifestyle. Endophytes, of course, mainly mycorrhizal species, many wood decayers and soil and litter decayers. So can we link these two? On one side, genomic traits, on the other side, ecological traits. And for that, in collaboration with the Joint Genome Institute, which is a branch of the US Department of Energy, with a sequencing center in California, in Walnut Creek, with the JGI, with a long-standing collaboration, which started, as Marcel emphasized a few minutes ago, by the publication of the first genome of a mushroom and the first genome of a mycorrhizal fungus, Lacaria bicolor, in Nature in 2008. Since then, we have sequenced more and more genomes, moving through the genome space of mycorrhizal associations, sequencing ectomycorrhizal species, arbuscular mycorrhizal species, ericoid or orchid mycorrhizal species. And today, lucky we are, because we have this month, we have more than 300 species of mycorrhizal symbionts, which have been sequenced by our group in collaboration with JGI, but also by many other teams. So we are our community can use now this outstanding genomic resource to get a better understanding of the biology of these fungi and their ecology. And I take this opportunity to mention that we are aiming to sequence, in collaboration with the JGI, we aim to sequence 1,000 mycorrhizal genomes. And so this is a call for applications. Anyone willing to sequence his or her pet mycorrhizal species, just please contact us and we will be very, very pleased to sequence these species. So what were the key conclusions that we gather from that large-scale genomic sequencing project? So now, almost 10 years ago, with undergrad Kohler, we published our first extensive analysis of the mycorrhizal genomes, focusing on ectomycorrhizal associations. And we demonstrated that by walking along the tree of life of fungi, here we are focusing on Basidiomycota, every time we sequenced an ectomycorrhizal species labeled ECM, like Lacaria bicolor, but also Cortinarius glaucopus, or Amanita muscaria, or many Boletales, every time we trace back the ancestry of that species, we were able to demonstrate that their common ancestors were saprotrophic species. So on every clade of ectomycorrhizal fungi, there is a saprotrophic ancestor. So this was really a major conclusion, emergence of ectomycorrhizal symbiosis from saprotrophic ancestors. And everything started about 180 million years ago when the Pinaceae evolved there. You see the Pinaceae evolution or emergence in the Jurassic. So we date back the ectomycorrhizal emergence through that time when the Pinaceae evolved. And we noted at a very massive radiation of ectomycorrhizal clades when the angiosperms evolved. So first key conclusion, symbiotrophs, ectomycorrhizal symbiotrophs evolved from soil or litter decayer or even wood decayers. And one of the most striking results was summarized here. This is a PCA showing the gene copy number for genes coding for enzymes that we call K-enzymes, carbohydrate active enzymes, those enzymes which are acting on cellulose, hemicellulose, pectin, lignin. And so by counting the number of genes for that enzymes, plant cell wall degrading enzymes, fungal cell wall degrading enzymes, we found that there were a striking difference between the different lifestyles. You have a series of clouds there, green, pink, gray, blue. These reflect the different cluster of species belonging to the different lifestyles. On the right side of the PCA, this is the lowest number of K-enzymes. On the left side of this PCA, this is the highest number of K-enzyme genes. And you see clearly that arbuscular mycorrhizal fungi here, they are lacking most of, if not all, enzymes able to act on plant cell wall polysaccharide. Then we have a big clouds of green there, where we found the ectomycorrhizal species. Most of them have a restricted set of K-enzymes. Still, some of these species are able to decay pectin or some glucans. But in any case, the number of genes coding for these K-enzymes is much, much lower than what we observed in the brown rot fungi, in the soil and litter decayers, or in the wood decayers. So we showed there that during the evolution of symbiosis, during the evolution of the ectomycorrhizal symbiosis, we have a striking loss of the genes coding for these enzymes. But it came to a surprise when we look at the gene copy number of K-enzymes for ericoid and orchid mycorrhizal fungi. We found here, you have the ericoid, we found that most of these mycorrhizal associates have a very large set of K-enzymes. And we can see that better on the following PCA, where I have summarized, I clustered the different genes for K-enzymes in the arbuscular mycorrhizae. Again, almost none, no genes for these enzymes. Ectomycorrhizal fungi here, a wide diversity, but most of them have a restricted set of genes coding for these degrading enzymes. But in contrast, orchids and ericoid mycorrhizal fungi have a very abundant repertoires of K-enzymes. So they have a dual life. Ericoid and orchid mycorrhizal fungi are able to form mycorrhizal associations, but they're also able to feed on their soil organic matter. So there we have a striking link between the ecology and the gene repertoires. We were very lucky to focus on the K-enzymes, on the K-enzyme genes, because this is really one of the group of category of genes where we have this very, very tight link between the ecology and the number of genes. Then moving to the biology of the system, we have been using the same genomes and the same gene repertoires to get a better view of the genetic landscape of these fungal tree interactions. We have used these genomes, gene repertoires, to understand what are the genes, what are the signals used by the mycorrhizal fungi to interact with the plants. I'm going to take only one example of that work. This is a work that we have been carried out on the small peptides, which play a role of effector. So this work has been done in vitro systems. So first we have developed these poplar Laccaria in vitro systems where we can produce thousands of ectomycorrhizae in a few days, synchronized development of the ectomycorrhizal associations. We can easily characterize the morphology of this association at the different steps. And then we were aiming to understand what are the signals, what are the molecules, what are the peptides exchanged between the two partners. So to make a very long story short, we have been able to demonstrate that the fungal hyphae here, which are landing on the root surface, is releasing in the medium small peptides, the red beads you see there, and some of them are able to enter the root cells and to very quickly translocate to the host nuclei, where they can change the gene expression of the host partner. And this has been very well demonstrated. The role of this mycorrhizae-induced small secreted proteins, MISSPs, has been nicely demonstrated over the last few years. Everything started by the work that Jonathan Plett carried out in our team more than 12 years ago now, and recently José Marquez Galvez published a very nice paper showing that MISSP7 is able to interact with the Jaz receptor here. These Jaz receptor are able to interact with a series of transcriptional factors like MYC2.1, MYC2.2, and those transcriptional factors are controlling the difference reaction in planta, and more specifically are able to control the expression of terpene synthase and the terpenoid metabolism, which are known to restrict the mycelial growth. So I would like to, you see, this is only one slide, but this is a work of a dozen of PhD students and postdocs, and it took almost 15 years to get there. Between the identification of MISP7, when we sequenced the Laccaria genome in 2008, and now that we have found the target of MISP7, it took almost 15 years. So, as you know, Marcel, science is not, it's really not a race, it's like a marathon. You know, you have to take your time and to develop a project on many years. And so if I summarize the data we gather from that large-scale genomics in a few takeaway points. So we have showed that the multiple evolutionary transitions from saprotrophy to ectomycorrhizal associations, and I think it's true also for arbuscular mycorrhizal associations, these transitions entailed first the evolution from soil, litter, wood saprotrophic ancestors, a widespread striking reduction of genes coding for degrading enzymes, those acting on lignin and cellulose. And we also found that many of these ectomycorrhizal species are lacking secondary metabolite clusters, so they are less able to synthesize toxins, for example. But we also found that adaptation to mycorrhizal symbiosis is not only losing genes, it's also co-opting genes which were already encoded by the saprotrophic ancestors, but in the mycorrhizae, thanks to new promoters, they can play new symbiotic functions. And clearly, many of the membrane transporters are regulated by symbiosis. We have found that ectomycorrhizae development and arbuscular mycorrhizae development is also entailing the formation of novel genes coding for mycorrhizae-induced SSPs, which are plant effectors able to dampen the defense reactions and to control the metabolisms of the plants, of the host plants. One point I didn't mention up to now is the fact that in all these genomes of mycorrhizal associates, we found a high content in transposable elements, much higher than in their saprotrophic cousins, relatives. And frankly speaking, we don't know why. And I think this is a very interesting field of research, why mycorrhizal species are accumulating so large content of TE is still a mystery for me. We found that all these mechanisms are taking place in many hundred of fungal lineages. It has been confirmed in the Agaricales, Boletales, Pezizales, in many fungal families. So we have a nice convergent evolution in hundreds of fungal lineages. And interestingly enough, we never observed a reversion from the ectomycorrhizal lifestyle to saprotrophy. So in fact, we have here, by looking at by using genomics to understand the evolutionary biology of these systems, we have found a very nice adaptation to a new lifestyle, how to live within plant roots, which is involving both gene loss, less enzymes or no enzymes, and a gene gains like the formation of new effectors. Of note, and this requires more work, we need to sequence more orchid and ericoid mycorrhizal fungal genomes, how these guys, which are able to feed on the soil organic matter, at the same time they are able to penetrate in the roots and to form mycorrhizal association. What kind of mechanisms they are using to have these dual lifestyles is really, I think, a project for the future. So now I would like to move on one of my favorite research projects currently. It's how we can use these genomes to follow gene expression, not only in an in vitro system, not only in a pot, not only in a greenhouse, but can we today use these genomic resources to investigate how these associations are working in situ, in the real world, in very old forests? Can we move to the field, collect samples, and get an overview of the gene expression landscape, transcriptome landscapes? And so we have published recently 2 papers which are summarizing the work of 2 very large consortia, one involving people in Canada, people in Sweden, in Spain, in France. And we have published this work in the Mycorrhizal special issue in New Phytologist, how to get to the bottom of fungal functions across a diverse set of forests. There in this paper, we have used meta-transcriptomics to shed light on the link between the functional traits of the different fungal guides, the saprotrophic species, the symbiotic species, the pathogenic species, and the ecological traits in different forest ecosystems, in the boreal forest, in the temperate forest, but also in the Mediterranean forest. So I just refer to that work, but I'm going to take another example of how we can use this large set of genomes to get a better view of the gene expression in forest soils. But before showing the results, let me emphasize that this was quite challenging. It took us almost 10 years to get there, 12 years. The project has been submitted to the JGI community sequencing program in 2011. So we had to solve many bottlenecks, many challenges, and the main limiting steps are summarized here. First, it was key to design efficient protocols for the extraction of soil RNA, the eukaryotic RNA. There are loss in a sea of bacterial RNA. So we had to design specific protocols, procedures to keep the integrity of this soil RNA and to fish them out from the soil matrix. Then, of course, when you have RNA reads, you can sequence now terabytes of RNA by using Illumina sequencing. That's fine, but then you have to identify from which species they are and what kind of function they are fulfilling. And so for that, you need to map RNA reads to fungal reference genomes. And we have been lucky enough, thanks to this collaboration with the JGI, to generate more than 2,700 genomes of fungal species from different ecosystems. And so now we can map about 50 to 55% of the RNA reads from a soil to this genome to these reference genomes. And I think this proportion will increase with time because we are sequencing more and more fungal genomes. And then, of course, you have to design specific bioinformatic pipelines to handle that terabyte of RNA sequences. So thanks to our very skillful bioinformaticians, we have been able, and technicians, we have been able to go through these bottlenecks. And I'm going to take one example of what kind of data we collect from that meta-transcriptome analysis. So this is an experiment which has been carried out in collaboration with Professor Yusheng Dai from the Beijing Forestry University, Zuliang Yang and Gang Wu from the Kunming Institute of Botany. And this experiment took place in a very beautiful place of the world, in the northwestern part of Yunnan in China, in a mountains called the Jade Dragon Snow Mountains, Yulongshushan. And here you see this white circle here. Here, at an elevation of about 3,800 meters, there are old growth forests, pine, spruce, fir, oak forest. And so we were able to compare the soil fungal communities of three of these forests: oak, spruce, fir, primary old growth forest. We've collected samples there, and we compare the taxonomic diversity of the soil of this forest, but also gene expression taking place in that soils. So this is a very smart team. Of course, you need very skillful people to work in these high altitudes. And just briefly, two main results. So we have extracted the DNA here from soil cores. We sequence them by using ITS, metabarcoding, in the spruce, in the oak, and in the fir forest. And here you have a heat map on the left part of the slide. You have a heat map summarizing the results. This is a OTU there in the three types of forest: oak, 3 replicates, spruce, 3 replicates, fir, 3 replicates. And the density of the color, very deep blue means high abundance, white means, or light blue means very few, very low abundance. And as hopefully you see clearly there, the fungal community in the soil of the oak forest is strikingly different from the fungal community here in the conifer forest. Instead, the spruce and fir forest are sharing very similar OTU distribution. So we have found here that, as expected, it has been showed already many times, that the tree species is impacting strikingly the fungal distribution and the fungal composition in the soil. The oak fungal community is very different from the spruce and the fir. Only 20% of the OTUs are shared between the three forests. So we have a striking taxonomic distribution. What about the functional diversity? What about the gene expression? So on the same sample collected in the soil there, of this old growth forest, we also collected and extracted mRNA from the eukaryote. We fished out the eukaryotic mRNA and we sequenced them on a large scale, several terabytes of sequences, and we mapped them to the fungal genomes at JGI microcosms. And so that's really, this slide is summarizing the transcriptome, the metatranscriptome landscape of these soils. If we move to the top left of the slide, you have the distribution of transcripts among the different lifestyles. In green, you have the transcripts from the arbuscular, very few, deep green there, very few arbuscular mycorrhizal genes are expressed. A substantial, between 20 and 30% of the transcripts of the metatranscriptome are from ectomycorrhizal species. But you see there that most of the transcripts are coming from saprotrophic species, either wood decayer or litter and soil saprotroph. And very little, very few of the transcripts here were expressed by pathogenic species. So in this soil, in the oak, in the spruce, and in the fir forest, in the organic layer or in the organic organomineral layer, we showed that most of the transcripts are expressed by saprotrophic species, even if symbiotrophs are representing a substantial part of that transcriptome. So we can really see there, we can assess the gene expression of the different fungal communities in different soils, in different soil layers. Now, if we move to the top right of the slide, this is the transcript coding for the translational ribosomal machinery, which is very often tightly correlated to the growth. And so this is some kind of expression of the growth-related transcripts. Again, in the fir in red, in the spruce in green, and in the oak forest in the topsoil and in the deeper soil. So we see there that we have clearly a higher expression of higher gene expression in the fir forest compared to the oak forest, and a higher expression in the topsoil than the deeper soil. So somewhat this is reflecting the growth of the fungal hyphae for these different communities. Now, if we focus on more specific functions, those which are involved in the metabolism, in the translocation of carbon compounds, nitrogen compounds, or inorganic ions, there on the bottom part of the slide, you see that surprisingly, there are the level of expression of these genes in the ectomycorrhizal symbionts or in the saprotrophic guides. Within each guide, the gene expression is very similar. We don't see a striking divergence in the gene expression between the three forest types. This is really in contrast with the taxonomic diversity I emphasized on the previous slides. And we obtained the same result for most of the gene categories. So here we see that at least for the transport-related transcripts, we have a very similar gene expression, some kind of functional redundancy within that functional categories. Now, this is not true for every gene categories. When we focus on the K-enzymes, again, those enzymes involved in the soil organic matter decomposition. And when we look at the gene expression, the total gene expression for these different K-enzymes, those acting on the fungal cell wall or those acting on the plant cell wall, in the saprotroph or ectomycorrhizal species, we see that the two guides are behaving very differently. The saprotroph, they're able to express genes for K-enzyme acting on the fungal cell wall. Very deep red mean strong gene expression. Similarly, this saprotroph, as expected, are also able to express a large set of genes coding for plant cell wall degrading enzymes. But if we look at the ectomycorrhizal species, within that guide of fungi, we observe that they have a substantial ability to decay chitin or fungal glucans through the fungal necromass. So this ectomycorrhizal fungi, they have kept a substantial activity to decay organic matter, but mainly the microbial necromass. Instead, they have lost, as I showed you in the previous slide, they have lost most of their ability to decay the plant cell wall polysaccharides and lignin derivatives. So by using this transcriptome data, we can really assess how the different fungal guides, saprotroph, ectomycorrhizal, pathogenic species, can act on the different substrates. So to summarize this part of my talk, I think we can say today that meta-transcriptomics can be used to survey gene expression in situ. We can go to the field, we can go to grasslands, peatlands, different types of forest ecosystems, plantations, old growth forest, and we have the ability today to assess the gene expression, the transcriptome of these fungal guides. And as we have showed here, in many forests we have investigated, we found a very diverse taxonomic diversity, very often striking shift between the different forest types, oak versus fir versus spruce. But despite this taxonomic diversity, highly variable composition of fungal OTUs, we have found that the main function, the core functions, those related to growth, development, sex, are very often very similar. There's a kind of ecological redundancy, or some people would prefer to say functional similarity, a striking taxonomic diversity, but a functional similarity. But still, we have found that by focusing on some group of genes, like the K-zymes, we have adaptation of the different fungal guides to their niche habitats, suggesting that there is some kind of niche adaptations. So, of course, this is only the first conclusion of a first series of experiments. I hope that thanks to the tools we have developed, thanks to the genomes we are sequencing massively today, more people will be able to use these tools to survey what's going on in the different ecosystems on the planet. So what in the future? What do I foresee for our community? Of course, today I'm talking mainly about genomics. So I think that we should collect more genomes. Today, even if we have 300 fungal mycorrhizal genomes sequence, it's only less than 1% of the 50,000 species of mycorrhizal fungi which have been identified so far. So there's a long way to go. We need to sequence at least 1,000, hopefully one day 10,000 genomes of ectomycorrhizal fungi to get really the true genetic and functional diversity of these fungal guides. I would like to convince our younger scientists also to sequence, to collect genomes in poorly known ecosystems. Most of the data we have today are coming from the boreal forest or the temperate forest. That's not enough. We need to go to the Arctic, in Antarctic. The tropical biomes should be explored to collect new species and hopefully to sequence them to be able to assess the functional diversity in these ecosystems. And I think that within the next 2, 3, 4 years, we are going to see more and more metatranscriptomes. And I think that by getting dozens of fungal metatranscriptomes, hopefully fungal metagenomes from diverse forest or grassland or peatland ecosystem, we will get a better view of the ecology of these mycorrhizal associations. And I want to couple that to the SPUN project. As you know, Toby Kears is leading a foundation, the Society for the Protection of the Underground Networks. We have now many underground explorers going through the world, the most remote part of the world, collecting samples in Patagonia, in South Africa, in Mongolia, in Siberia, many places where we've never been. They are collecting samples to assess the fungal diversity. That's great, and we should really fully support that program, and hopefully we can explore many more ecosystems. But I would like to suggest to couple that a survey of the fungal diversity, taxonomic diversity. We should carry out metatranscriptome analysis surveys in all these spots where we collect samples for the fungal taxonomy, to assess the fungal diversity. Hopefully, we can also collect enough samples, RNA, to assess fungal function. And so by doing so, we may, I hope, in the few years, we may have an atlas of the metatranscriptome, a global one at worldwide metatranscriptome atlas. And then we can use that information on the diversity, on the functioning, to popularize science with the local communities. We can use this information to educate school kids, but also other people, politicians including, and then we can be able to lobby the different political systems to protect fungi, because climate change is really today impacting massively this fungal community throughout the world. Okay, I think it's time now for me to summarize, to thank many people. I mean, the work I really today I talk on the behalf of many groups, including my team, our team here in Nancy, with Marc, Annegret, Claire, many skillful bioinformaticians, Shingo, Luca, Emmanuel, Laure, who has been able to design specific protocols for RNA extraction of soils. Jose and Gaurav, who did great work on the MISSP7 program, and my colleagues in China, Fong, Annie, Gang, Chinchao, and I should cite my long-standing collaborators, Igor Grigoriev at the JGI, Bernard Henrissa, the master of the K-zymes, David Ibet, who is an amazing phylogenomicist, and Laszlo Neich, who is working with us for a long time on the developing new tools for mining these genomes. And with that, I think I'm done, and I'm ready to answer some of your questions. Thank you very much.
Yeah, thank you very much. Really fantastic talk. Very interesting. New ideas again. The atlas for metatranscriptomics. It's an obvious thing to do, but it would be really cool to see these data in a couple of years, I guess. So yes, I think the floor is open for questions.
Maybe you can type the questions, and then I can try.
Yeah.
I see the chat is there. Don't be shy.
Yeah, so maybe we can, Daniel, if you want to do directly your question.
Hi, hello. Can you hear me?
Yes, I do.
Okay, so congratulations for the presentation. It was great.
Thank you.
We follow your papers. They are amazing. So thank you for the presentation. I saw this some of your papers, especially this one from Our, that's about transcripts of fungal guilds. There are a lot of discussion about the ECM transcripts, but we don't see much about AMF. There is just one line in the paper, but don't talk much about AMF. And you mentioned something about challenges to get more mRNA from soil. So what are the challenges that we can see better the transcripts from AMF, and what's known about AMF in soil, about the transcripts? Thank you.
Yeah, good points. In fact, in the forest, we have been investigating in the boreal temperate forest in Quebec, in Sweden, in Spain, in France, and the work we did on the Chinese in the Chinese mountains in Yunnan. Most of these ecosystems have very few arbuscular mycorrhizal trees. Most of the samples we have collected were from ectomycorrhizal plants, host plants, or ericoids, but very few arbuscular mycorrhizal systems. So I think that if we move, even when we move to the meadows at more than 4,000 meters high, in these meadows of Yunnan, most of the species are sedges, and these sedges are ectomycorrhizal species associated to Cinococcum. So I think if we want to get more information on the metatranscriptome of arbuscular mycorrhizae, we should use them in meadows in the European Alps, alpine meadows, for example, or any grasslands, pastures, So I don't think it's because we missed the RNA reads. I mean, it's just because in the soil samples we have investigated so far, arbuscular mycorrhizal species were very lowly represented. Because I think there's enough genomes now. There are more than 12 genomes of arbuscular mycorrhizal fungi at the JGI. It should be increased, of course, but it's still enough to map most of the arbuscular fungal species. So I think it's just a question of low percentage of arbuscular fungi in the ecosystems we have analyzed. But I know that there are 2 or 3 programs in Europe where people are starting to use metatranscriptomics to investigate pastures. For example, the experiments that Marcel is developing in Zurich, I'm sure that metatranscriptomics will be very useful to survey the function expressed by arbuscular mycorrhizal fungi.
There is here one question. You also see it, I think, given that mycorrhizal fungi from Olivier, I think, Olivier Herlin, given that mycorrhizal fungi do not have many of the enzymes to degrade plant cells and fungal cells, how do ectomycorrhizal and AM depend on if the arbuscular mycorrhizal fungi and AM depends on subtrophic fungi for nutrients? So not only the mycorrhizal helper bacteria, but also it says subtrophic fungi.
I would think that I would say that in fact the main carbon provider for arbuscular mycorrhizae and ectomycorrhizal fungi, as you probably know, is the plants. So if my speculation would be that when the ectomycorrhizal associations evolved, they relied more and more on the plant glucose to feed and to develop, and then step by step they lose their repertoire of lignin and cellulose degrading enzymes. So they have lost these enzymes because they are fed by the plant's glucose. Instead, for nitrogen, I think that we have found that many of these ectomycorrhizal associates, and likely some of the arbuscular mycorrhizal associates, are able to degrade peptides and proteins. So when the mycelial networks are colonizing the organic matter, the litter, the soil organic matter, they can probably use some of these enzymes to scavenge nitrogen. And the group of Anders Tunlid in Lund has nicely showed that these ectomycorrhizal fungi, they have lost their ability to decay cellulose and lignin, but they have kept a very striking ability to decay peptides and organic nitrogen in soil. So they can compete with the saprotrophic species in that kind of niche habitats. So what else? We are working with RNA-seq of AMF and retrieve only 0.5% of the AMF sequences. Maybe also the biomass of EMF is so low. Yeah, of course, yes. I think that it was really a challenge in our forest soils to collect enough mRNA from eukaryotic organisms, including fungi. So we have to design specific protocols based on oligo-DT tail to really select the eukaryotic mRNA from the ocean of RNA from bacteria. So it's likely similar issue for your experiment on AMF.
So, Francis, Vinit Vishal from India says, our eDNA samples have got few DNA sequences, less than 0.1 million reads. The sequencing company always said our data has some metabolites, which could be the problem for isolating and sequencing. or the environmental samples. So I want to ask how we can deal with them.
As I say, it's really a very special recipe. I mean, we have, of course, you can use commercial kits, and we are using Molbio Power Soil Kit to extract RNA, but you need to provide additional protection to the eukaryotic RNA. So it's not, it remains challenging to extract RNA, and it requires optimization of the protocol for each type of soil, for each type of ecosystems you are dealing with. You may need to optimize the procedure to use to include additional protection to get enough RNA. But I think that you say 0.1 million reads. I think we sequence more than 100 million reads per sample. So I think probably it's not enough. You need to do a lot of sequencing to get a good picture, a good image of the transcriptome. So as I mentioned in my talk, we in collaboration with the JGI, we sequence more than 1 terabyte of RNA, so probably more than 100 million reads per sample. So it's still one major limitation of metatranscriptomics is still the budget. It's still very expensive to do Illumina sequencing. And if you need to do that on 100 of samples, very quickly, the budget is rocking. Yeah.
Many thanks. So we have a last question. It says, Rodica Pena, you show that despite a high taxonomic variability, there is a stable community functional structure. Do you expect that this will change under a stress factor?
Yeah, good points. I would say that the main, again, that's pure speculation to me. Functional similarity or functional redundancy is probably a system to cope with stress. So I think that when this fungal soil community will experience drought stress or fire, many species will disappear, but there will be still many species surviving. And since they share the same function, the ecosystem should be less disturbed by the stress. So I mean, it should be tested. We haven't done any experiments up to now. It would be very interesting to test the impact of drought stress on the metatranscriptome. It should be done. But I would think that at least the functional similarity that we have observed is probably helping the fungal community to cope with many stresses. But it's your duty now to check that.
Okay, so we are going to close our seminar for today because our Zoom link will expire soon, so we cannot take more questions, unfortunately.
People can send me the question by email or through the IMS website, and then I can try to provide some feedback.
Awesome, thank you very much. Because there are a couple of questions more, so they can write you directly, right?
Yes.
IMS. Okay, so we would really like to thank you, Francis, for your excellent presentation, and of course to all of you to for attending and make this amazing question as well. We will also like to thank to Marcel for this nice introduction of Francis and for the discussion, Anna for the logistical help. We want to also thank to Professor Cesar Marin at the UST in Valdivia, Chile, and guest researcher at VU Amsterdam for supporting us today with the Zoom platform for this seminar. And before to leave, I want to do a couple of announcements, if you allows me, that might be of your interest. So I will share my screen very quickly. I guess. Can you see my presentation? I believe so. Yeah. So we want to invite you to follow IMS social media, because we will be uploading this seminar to our new YouTube channel. So we also have the Twitter and Instagram, so you can follow what we are doing. And of course, to know a little bit more about, and you maybe repeat this seminar through the YouTube channel. Also to invite you to our conference, the ICOM, that will be in Manchester. It's very soon now. So please, we will really like to see you there. Also, please consider this interesting event in Brazil about ecological and evolutionary consequences of plant fungal invasions. We want to invite you also to review our nice newsletter that is led by Cesar Marin as well, and it has many interesting features as the top 10 papers on mycorrhizal research. You can check them in our webpage. And also invite you one more time to read this latest special issue that Francis Martin mentioned a lot in his talk, and also because all the articles are free during May. So I really want to invite you to take a look of this amazing collection of mycorrhizal research in new phytologists. So that will be, oh, sorry, to stop to share. So there were my announcement. And once again, many thanks, Francis, for being here, for giving us the first talk. We really admire your work. So for us, it's also a pleasure and an honor to have you here with us.
Thank you, Patricia. Thank you, Marcel.
Yeah, so stay tuned, because we will have the second seminar at the end of June. We will have very nice speakers, Matthias Rillig and Vasilis Kokoris. So stay tuned for detailed information on the date and the time. And we will looking forward to meet you there again in this new IMS seminar series. So see you then. Bye-bye to everyone.
Thanks a lot, Patricia. Great that you organized this. Thanks.
My pleasure, Marcel. See you. Bye-bye. Let me stop record. Okay, bye-bye.